to introduce Dr. Nelson Ting. So Nelson is a new faculty member in the Department of Anthropology here at the University of Oregon, whose research interests align quite a bit with many of us in the Institute of Ecology and Evolution. So Nelson received his PhD in anthropology from the City University of New York Graduate Center, where he actually worked in the New York Consortium in evolutionary primatology. Right. Before that, he obtained an MA, a master's degree from the University of Missouri Columbia, and a BA in biology and anthropology from Washington University. Many of you know one of the best places in the world for a combination of anthropology and evolutionary biology. And Nelson is really interested in the intersection of studying anthropology and evolutionary biology and ecology, especially using primates. So his interests are in molecular ecology, evolutionary biology, and some of the major questions that drive his research are understanding the biological and social drivers of primate disease transmission, actually how do ecology and evolution play into how diseases are transmitted, the role of climate change as it affects primate population dynamics, and then primate molecular systematics and primate social systems and behavior. So Nelson's actually going to talk to us a little bit today about some of that research, and the title of this talk is Identifying Conservation Priorities and the Effects of Environmental Change for Africa's Most Threatened Primates.
All right, thank you for the introduction, Bill. I've met many of you in this room, but I realized that I've never actually talked here, so a lot of you don't really know what I do. So this is a great chance to tell you about my research. So thanks for inviting me. So, like Bill said, I am a molecular ecologist, or that's typically how I would self-identify. But I'm also a biological anthropologist. And as a biological anthropologist, by definition, I do have an organismal focus. Biological anthropologists are interested in not only human evolution, but also the evolution of our closest relatives, the non-human primates. So we've, for a very long time, realized that non-human primates can serve as a very good model for understanding our own evolutionary origins. So we share a lot of the same characteristics. Large brains, relative large brains, long life history, high degree of sociality, and the close phylogenetic relatedness allows us to use non-human primates as a good model for human evolution. So another reason to study non-human primates is, ecologically speaking, they tend to be a good indicator species. So they're very sensitive to habitat disturbance, and because of their long life history, they take a long time to recover from population declines. And from a conservation standpoint, because they're our closest relatives, they are very charismatic taxa that draw a lot of conservation interest and funding. So these are several of the reasons why biological anthropologists study non-human primates, and as a molecular ecologist, I'm interested in addressing questions using genetics involving the evolution, ecology, and behavior of these organisms. So when I just mentioned habitat disturbance, and I say primates are, or many primate species are very sensitive to habitat disturbance, what exactly am I talking about? One of the probably the greatest long-term threat to non-human primates is deforestation. So this makes sense. If you cut down an animal's habitat, or you destroy their habitat, then they will die out. So although many primate species have gone on to exploit subtropical environments and non-forest environments, or some have, the vast majority of primates are still adapted to this kind of fundamental ecological niche, these tropical forested ecosystems. So whether it is deforestation for agriculture or cattle pasturing or logging, this is the greatest long-term threat to these animals. So this is probably one of the more dramatic examples of deforestation that you can find. These images are showing how much forest has been lost in the islands of Sumatra and Borneo in Indonesia and Southeast Asia. So these are where orangutans, one of our very closest relatives, are endemic. So you can see how much forest has been lost in these areas and how much is projected to be lost out to 2020 in this case of Borneo. And certainly, very likely within our lifetime, we'll see the extinction of Sumatran orangutans in the wild. they only exist in this upper corner in this forest fragment currently. So it's just a very dramatic example of the threats that deforestation have, or the threat of deforestation on wild primates. The other major threat is hunting. So as you know, being adjacent to these forest communities, or even part of these forest communities, are human populations that extract resources from these communities, a lot of times using primates as a protein resource. And this has become a large commercial trade where you have, at least in Africa, it's known as the bushmeat trade or the bushmeat crisis, where you have bushmeat traveling international boundaries, even going to places like Europe and the US to immigrant communities who find this food as comfort food. And it's important to point out that while I mentioned how the greatest long-term threat to primates is deforestation, hunting can actually extirpate primates from habitat much faster than deforestation can. So there are examples, for example, in Ghana, of this case called empty forest syndrome, where you have these very large forests that are relatively undisturbed in terms of habitat destruction, but they're very empty. They're devoid of medium to large-sized mammals, including primates. So because of those threats, a lot of the work I do, this molecular ecology work, has a lot of implications for conservation biology. So one of my interests is in conservation genetics. So when I asked Bill and Brendan exactly what should I talk about for this talk, they said, to give people an idea of who I am. So I'll do this by following this outline here. So I introduced some of my interests in primate molecular ecology and some of the threats to primates and how my work, so you'll see kind of how my work intersects with that very short introduction there. But what I really want to talk about is some of my recent work and recent interests in environmental change. So understanding how environmental change affects primate demography, both in the past, in the present, and what the implications are for the future. But before I do that, I kind of want to go over some of my past work, just to give you an idea of what I've done already, how that interrelates with what I'm doing now, and to give you an idea of what some of my interests are. I have some pretty broad interests, so this will give you an idea of what I've done and what I'm planning on doing in the future. So this past work involves columbine monkeys, their evolutionary history, and some of their behavior and ecology. So what are columbines? These are old world monkeys. They're found in the tropics and subtropics of Africa and Asia. And they belong to the same family as the Cercopithecines, or the cheek pouch monkeys. You're probably more familiar with those, things like macaques and baboons. And a lot of those animals, like macaques and baboons, are used as biomedical or evolutionary models for human origins. But very little attention has been paid to columbines, even though they're just as closely related to us as that other group of monkeys. So I would argue that by understanding aspects of columbine evolution history, their behavior, their ecology, we can further understand, or it can give us insight into our own origins as well, just as studying things like macaques and baboons have done the same thing. So I've worked on both continents here, both Africa and Asia. So the first thing I want to talk about is some of the work I've done in Asia. So let me just go back one slide. So there are two interesting groups of Asian columbines here. You have these things called odd-nosed monkeys, so aptly named. They have these very odd noses. This nasalis monkey has this very pendulous nose. This snub-nosed monkey has this very smushed-in nose. And they're kind of a phylogenetically cohesive unit, these odd-nosed monkeys. And the other group of Asian columbines are the langurs and leaf monkeys. So these are separated into now 3 different genera, Semnopithecus, Presbytus, and Trachypithecus that you see here. And there have been some questions about how these 3 animals are related to one another. Morphologically, based on pelage pattern, and specifically neonatal pelage pattern, people have inferred a very close relationship between Semnopithecus and Trachypithecus to the exclusion of Presbytus. Now, interestingly, in 2006 or so, this mitochondrial paper came out. This was actually first authored by Kirsten Sterners, who some of you might know. She's now faculty in the Anthro department as well, in the crowd somewhere. So this work showed using the protein coding regions of the mitochondrial genome that actually Presbytis and Trachypithecus are very closely related, mitochondrially to the exclusion of Semnopithecus. So you had two kind of competing phylogenetic hypotheses here, one based on pelage patterns and one based on mitochondrial DNA. So what I did was I sequenced some of the X chromosome, about 4,000 base pairs of XQ13.3. And for this relationship, showing that actually, at least on the X chromosome, the close relationship between Semnopithecus and Trachypithecus is supported. So showing that, at this nuclear marker, the morphological hypothesis was supported. Now, since then, several other studies have been published, some by myself and my collaborators, looking at other markers across the nuclear genome, across sequence markers, across the autosomes, and also transposable elements. And really, if you look across the nuclear genome, this relationship is always supported. And what this is because of these branch lengths, divergent states, a really recent divergence between Semnopithecus and Trachypithecus in the nuclear genome, and a very older divergence in the mitochondrial genome between Trachypithecus and Presbytis, you're seeing a pattern of, or what most likely explains this pattern that you're seeing is ancient hybridization. So if you had sex-biased and unidirectional gene flow from Semnopithecus into Trachypithecus, so Semnopithecus males outcompeting systematically Trachypithecus males for female mates, and then over several generations, that would eventually lead to this pattern that you see here, where you have cytonuclear discord in these two parts of the genome. Now, ancient hybridization in primates, at least in biological anthropology, has always been of interest, especially recently. If you notice, the publication date of this was 2008, so this is before the Neanderthal genome was published. There was a lot of questions in human origins about whether Neanderthals and humans had hybridized before, and so there was a lot of questions regarding, how prevalent is ancient hybridization in primates, and what can this tell us about human origins and Neanderthal and modern human relationships. So this was kind of an interesting work showing that you do see ancient hybridization in primates. And now we do know that actually there was ancient hybridization between Neanderthals and humans now that those genomes have been sequenced. So this is some work that I did on the Asian side of the Colivine tree. Now some work on the African side involved these things called colobus monkeys. And this is the last African primate action plan developed by the IUCN. So to designate conservation priorities for all of African primates. And on the cover, what you see is not something you might expect to see, like a chimpanzee or a gorilla. What you see is this thing called a red colobus monkey. So red colobus monkeys are some of the most endangered primates in the world. This is their distribution across the African rainforest belt. They're distributed allopatrically in these 16 to 18 different forms, allopatrically except for this one hybrid zone that you find in Central Africa. To give you an idea of how threatened these animals are, numbers 1 and 2 are endangered. Number 3 is probably extinct, hasn't been seen in several decades. 4, 5, and 6 are either critically endangered or endangered. Number 7 is either is critically endangered or most likely extinct. It's not been seen in several decades. We don't know what's going on in Central Africa. It's very hard to get good estimates on population size and to get a good idea of how well protected the forests are there, but we do know hunting is rampant and poaching is rampant in protected areas in Central Africa. 15 is endangered. 16, 17, and 18 are all either endangered or critically endangered. So one of the problems about designating conservation priorities for this group is we don't know how all these different forms are related to one another. We don't know which ones might be closely related, which ones are distinct. And if you're trying to do conservation and preserve biodiversity, you really want to preserve evolutionary history. So you don't want to lose any kind of lineages that are phylogenetically distinct. So I sampled across this range and across almost all these forms, and using a combination of non-invasively collected samples, fecal samples, or museum specimens, I inferred this tree. So this is based on about 1,000 base pairs of protein coding mitochondrial DNA. And you can see there are 7 different phylogroups here in this red colobus tree. And if you plot these phylogroups on a map, you get this distribution. And this is interesting because prior to this work, most of the conservation efforts were really focused on East Africa, on these populations over here, especially these fragmented populations. And that's where people thought should be the highest conservation priority. But according to this work, if you really focus your efforts here, you'd really potentially lose out on a lot of evolutionary history and a lot of phylogenetic diversity that you find in West and Central Africa. And particularly G and C, these two lineages here, which have a very restricted geographic distribution. They suffer from habitat destruction and from hunting. And if you really kind of neglect these guys, you could lose a major piece of evolutionary history across this group. So this work was really important in elevating the Niger Delta red colobus, that's G over here, to one of the 25 most endangered primates in the world. This was just discovered at some point in the late 90s, and it's already critically endangered. And from this work, you can see it's a really kind of phylogenetically distinct taxon. Some other work that I'm working on with colobus monkeys. This is actually work being conducted by Ava Wickberg. So this is a graduate student that I'm co-advising with Pascal Sacut at the University of Calgary. And this is focusing on this group of black and white colobus monkeys in Ghana. So in this one part of Ghana, in these series of forest fragments, these monkeys, for whatever reason, because of local taboos, are considered sacred. So they're actually doing quite well here. And what we're interested in looking at here is the ecology of dispersal and patterns of kinship across these fragments. So basically understanding how ecology affects social behavior and why you might find different patterns of dispersal in different species. So these black and white colobus monkeys are a nice model to study this because you have different species across Africa that show different social systems. So you have some species that are predominantly female or very strongly female philopatric and male dispersed. You have other species that show no bias in sex dispersal, and you have this species which shows something in the middle, where you have predominantly male dispersal, but sometimes females disperse, although most of the time they do show philopatry. So why do you find these patterns? Why might you have variation in a social system in terms of patterns of dispersal? What implications does that have for kinship? Do you have groups where you have females who are socially bonded but not related? Or if females are dispersing, why are they dispersing? And are they dispersing to groups where they're finding closely related females? These are the types of questions we're interested in addressing. Ava's writing up her dissertation now, and we've kind of just submitted, we have some results. We've just submitted some of the work on the dispersal aspects of this research, showing that sometimes females do indeed disperse, and we're working out some of the different aspects of the social system, and why females might be socially bonded, and if kinship is, how much of a factor kinship is playing in social bondedness. So that's some research that we're trying to wrap up at the moment. So that's a little bit about some of the past work I've done on columbines, some of the work I'm trying to wrap up. And so what I really want to focus now on is some of this recent work. This is a paper that my colleagues and I just published in Wiley's new online journal, Ecology and Evolution, on this animal known as the drill. So I'll talk a little bit about the drill in a second, but really looking at how climate change has affected the demographic history of this animal. So why climate change? I don't think I need to talk too much about this, but clearly climate change, the idea of climate change has become very pervasive in our lives. You see it in legislature, you see it in commercials, you hear a lot about animals being affected by current climate change. So it's really become, like I said, a pervasive part of our lives. So understanding the effects of climate change is very important to the future of biodiversity. Now, many different researchers have done this by looking at how past climate change has affected animals to get a better understanding of how future climate change might affect them in turn. So these are studies done using coalescent theory. These are analyses known as skyline plots. And what you're seeing is effective population size on the x-axis, on the y-axis, and then time on the x-axis. And using coalescent theory and looking at the shape of the genealogy as it goes back in time, as alleles coalesce back to one ancestral unit, you can test whether the shape of the genealogy conforms to a model of population growth, population decline, or constant population size. And if you have a good idea of the mutation rate and of generation time, you can also time these changes in population size. So that's what these studies have done, and correlated these changes in population size with past climatic events. So this is work that's been done in muskox, the American pika, these now extinct European cave bears, and bison. So what all these studies have in common is that they're really looking at animals that live in northerly latitudes, Arctic regions, or alpine regions. So very little work has been done like this on animals that inhabit tropical forested communities. So I'd argue that this is an issue because if you look at some of these models that have been produced, so this is a paper that was published last year in PNAS, modeling where we're going to have the greatest amounts of climate change in the near future. This is, I think, over the next 80 years or so. And if you look at the red, that's where the most climate change is expected, and this is all in the tropics. So basically the tropical forested regions of South America, Africa, and Southeast Asia. So if we're really going to understand what the effects of this climate change are, we really have to understand how these past climatic events, how past climate change has affected animals living in these regions. So to address that, so this question: how has climate change affected tropical forest-adapted species? My colleagues and I looked at these specific forests in West Central Africa, known as the Cross-Sanaga-Bioko forests. Oops, sorry. Okay, so ignore the green for now, if you can, but these are the forests between the Cross River in Nigeria, the Sanaga River in Cameroon, and on Bioko Island, which is part of Equatorial Guinea. So this is an area of very high endemism. So this is one of World Wildlife's 200 ecoregions that they've designated as high conservation priority. And it's a very contained biogeographic unit. A lot of the taxa found here don't exist elsewhere. And right in the middle of this region, you have this lake called Brambi-Mbo. And the reason why this is a good model for looking at the effects of past climate and environmental change is we have a very good documented record of forest change because of the fossil pollen record at Brambi-Mbo. So this is one of the best records we have in the world for a tropical forested region over the past 25,000 years. And what you're seeing here, you have the modern day here, you have the percent pollen, so you have trees, you have forest, you have aquatic pollens, and you have savanna, and then time on the x-axis. You see that forest was decimated during the last glacial maximum. We had a glacial advance. Around 12,000 years ago, you have forest advancing during an interglacial period. And what I really want you to focus on right here is this time period around 3,000 years ago, where you have forest reduction, but in a warm interglacial period. And what happened here is, for whatever reason, you had an increase in aridity and seasonality in this region, which, even though it was a warm climate, because of that aridity, you had this forest reduction in the area. So if you kind of keep that in the back of your mind, that'll become relevant in a little bit. So like I said earlier, the animal we focused on is known as the drill. We focused on this animal because it's a large-bodied mammal, so we wanted to know what the effects of this past climate change was on this type of animal. Also, it's heavily restricted to a tropical forested environment, so you don't find it wandering off into the savannas on the edges of this primary forest. And also, from a conservation perspective, this is a very, very endangered animal. So very little is known about the drill. It suffers from habitat destruction and poaching. It's very cryptic. We know very little. We know some. We're starting to know a little about its ecology, very little about its behavior, and pretty much nothing has really been done on its genetics until now. And in fact, in the African Primate Action Plan that I showed earlier, the drill was considered the highest conservation priority for African primates. So this is the geographic distribution of the drill. You see in green. That's where it exists in the forests, in the Cross-Nago-Bioko forests. And the stars are areas that we were able to sample. So the sampling was not comprehensive in terms of locality, but this was really the best we could do. This is not an easy animal to sample from. But what we were able to do is we were able to sample its presumed largest populations in its largest forests, and also the kind of extremes of its geographic range to get an idea of, to try to maximize the amount of genetic variation that we were capturing in this animal. So a lot of my work does involve field work, and the kind of details of the field work involved in these different sites really vary. But I thought I'd just give some examples of what the field work, or one example of what the field work is like. This is the island of Bioko. So like I said, this is part of Equatorial Guinea. The capital of Equatorial Guinea is Malabo, in the northern end of the island. So I went here a few years ago looking to collect drill samples. And on this island, all the primates really are, they're highest in abundance, and they're really found, not only found, but really much easier found over here in the southern part of the island, which is very remote. And as you can see, there's a good reason why they're found here. It's because there are no roads there, so it protects them from hunters. So this was a very atypical trip in that I was part of a large expedition. So this is the Bioko This is an expedition that the Bioko Biodiversity Protection Program does every winter. So this is a group of researchers, students, and volunteers who go to Bioko to get these kind of, well, the volunteers and students are getting some wildlife experience with conservation. And the researchers are looking at anything from doing surveys of sea turtles or primates to people collecting samples like myself. So a lot of kind of interesting research going on here on Bioko. So this was a basically a 3-week trip into the forest where we were camping. Like I said, it's pretty remote, so you have to take a boat from Malabo, a 3-hour boat ride down to the southern part of the island. You can see some dolphins here playing in the wake of the boat. And then we set up various camps along the southern part of the island, and I moved around these camps looking for drills and collecting samples. Here's some Ecuador-Guinean students collecting some data, some student volunteers eating some sardine spam and sardine spam stew, I guess, was our staple dinner. This is a view from what is known as the Grand Caldera on the southern part of the island. So this is a volcanic island. So this is a volcano on the southern part of the island, and in the caldera, in the crater of this volcano is where the primates are most abundant. Because again, this is where it's also where it's most remote. So getting here involves a 7 or 8 hour hike up this volcano and then down into the crater. So this view doesn't really do it justice, but basically you're surrounded 360 degrees by this crater wall. So it's a very rewarding place to do work because it's a really beautiful place, but also very challenging. But this is where the primates were most abundant. So the drills here don't exist on their own. There's a pretty diverse primate community here. In terms of the diurnal primates, you have the drill, which I've already introduced. You've heard a little bit about red colobus monkeys and black and white colobus monkeys. And you also have 4 different species of guenin. So these are arboreal monkeys that you find throughout Africa. So you have 4 different species, closely related species there on Bioko. Now, as far as sample collection, I've already mentioned I've done a lot of work with non-invasive samples and fecal samples in particular. So that's more or less what I'm doing. I'm following these animals around, collecting fecal samples. When you work with endangered primates, especially ones that are not habituated and they're hunted, it's really impossible to do any kind of invasive sampling. So what we're really limited to when dealing with these types of taxa are these non-invasively collected samples. So following around these animals and collecting their feces. So from those fecal samples, I was able to amplify and sequence about 2,000 base pairs of mitochondrial protein-coding DNA. And the first type of analysis that we did was looking at population structure. So this is because if you're looking at demographic history and past population dynamics, at times if the certain aspects of population structure can confound demographic analyses in that they can mimic, for example, a population decline. So we wanted to make sure that when we infer the demographic history, we're not inferring a false history here. And what we find here is extremely strong levels of population structure in these mitochondrial lineages. You see very high FST values. You can see this haplotype network here shows that they don't share many haplotypes across these different localities. And you can see that displayed here in this mitochondrial gene tree as well. So inferring a false population collapse is problematic when you have mild levels of gene flow. It's not as problematic when you have no population structure or very strong population structure. So what you're seeing here is evidence of very strong population structure. And this is this Bayesian skyline plot that we obtained from the program BEAST. So we had good, we were able to obtain a mutation rate because we have a good fossil record for a closely related group in the baboons. And we also have a good estimation of generation time because there is a free-ranging captive colony in Nigeria where we have some life history data. And what you're seeing here is a 15-fold decline in female effective population size. So we have female effective population size, female because we're looking at a matrilineal marker on the y-axis and time on the x-axis with the modern time right here. You see a 15-fold decline in female effective population size in the drill. And the sharpest part of this decline is where you have that forest contraction around 3,000 years ago, where you have this very warm climate along with an increase in seasonality or aridity. Now, in terms of implications for the future, it's always very tenuous to predict the future, but that situation is potentially analogous to what we might see in tropical forested regions in the future. If we're in an interglacial now we have a relatively warm climate. The temperatures are increasing in these areas. But then also, because of human disturbance in the environment, particularly cutting down forests, we're disrupting the evapotranspiration cycle. So a lot of projections are seeing or are modeling the effects of these activities and showing that we could have a very warm and increasingly dry climate in these tropical forested regions. So if that is the case, then something like the drill would find a really hard time dealing with that type of situation. So in terms of conservation, protecting large areas of forest, so that would help preserve the evapotranspiration cycle. That would help in protecting habitat in the immediate term for the drill. And if you protect it effectively, you can protect it from poaching. That will really help this animal recover and from this population decline and also from any kind of recent disturbance, specifically hunting that it sees now. There were some concerns in terms of, I just want to show this very quickly, in terms of seeing how robust this demographic result is, about unequal sample sizes between the different localities, and then also if you analyze each locality individually, are you seeing this demographic decline? And so we did a resampling analysis as well. And no matter how we resampled, whether we resampled the localities with fewer individuals to get those sample sizes up, or whether we randomly took out individuals from the populations with fewer individuals, or whether we analyzed each locality individually and resampled to increase sample sizes, we always got this robust demographic result of a population decline occurring around 3,000 years ago. So we did quite a bit of work of trying to figure out, trying to make sure that what we're seeing is real in terms of this population decline. One particular issue I had with this is that the resolution here is not great, and it's been shown that when you use a single marker, a single marker is very limited in revealing demographic history, especially if your most recent demographic event was very severe. You lose any kind of signal of past demography prior to that event. So this is a sample. This was a study done by Held and Drummond in 2008, showing the true demographic function here is in blue, and showing how these skyline plots work with an increase in sample size but one marker, or a decrease in sample size but with more markers. And it shows that with more markers, you can actually get much better resolution in inferring what was going on further back in time. So this is something that I'd like to start doing, and using not only looking at the drill, but other animals who are part of those primate communities as well, to see, you have a very good idea of how the forest has changed. So they get an idea of what happened back in the Pleistocene during periods of climate change. And what, if you go back a couple slides, what was going on back here, which it seems that this demographic event here has probably wiped out the signal that occurred back here, where you have a lot of forest change as well. And a lot of that will be methodological development, trying to figure out ways to get multi-loci data, multi-locus data out of non-invasive collected samples. So that's something that I'm looking to work on as I move forward here at Oregon. Okay, so what I really want to introduce now is the current work that I'm focusing on habitat alteration. I don't have any results here because this is a project where it's a 5-year project where we just finished our first year of funding. And it's a project funded by the Ecology and Evolution of Infectious Disease Program, which is a joint NSF-NIH program. Now, there are multiple aspects to this project, but this is the main question that I'm interested in. But how has habitat fragmentation affected pathogen transmission and persistence in primate hosts? And we're looking at this primate community in Kibale National Park in Uganda to address this question. So we're using, again, this red colobus monkey model to address this question, but we're really interested in community dynamics here. So in this community, you have multiple different primates, about 13 different primate species. You have various plants and trees in this community, some of which provide food resources to these different primates. You have various predators who find these primates as prey. Some are primates themselves. Chimpanzees are known as a major predator of red colobus monkeys. You have parasites and pathogens, which these primates host. You have various other wildlife which may interact indirectly with the primates. And then, and then you also have humans. So you really can't understand these wildlife communities without taking humans into account. Humans have not only altered these communities in a way that you need to take that alteration into account to understand what's going on, but also humans are themselves part of these communities. So what I really want to focus on are these 3 players. So humans, this red colobus monkey, and also this virus known as SIV. So we're interested in looking at various retroviruses, but this is really focusing on simian immunodeficiency virus. So there's a lot of interest in SIV, or there has been a lot of interest in SIV, because once if certain strains of SIV jump into human populations, it is then known as HIV, which then often progresses to acquired immunodeficiency syndrome, or AIDS. So since the 90s, a lot of work has been done on the evolutionary history of SIV in the hope that understanding how this virus operates in wild populations and how it's evolved can give us some understanding into treatments in humans. So this is really one of our best characterized diseases of zoonotic origin. So just to give you a little idea about what we know about SIV, we know that almost all African primates, at least monkeys and apes, carry some kind of strain of SIV. But interestingly, for the most part, they don't get sick. They don't progress to any kind of AIDS-like symptoms. Now, this is something very specific to African primates and very specific to the continent of Africa. If you infect a macaque, which is native to Asia, with SIV, they will progress, or at least with HIV, or the SIV strain that is associated with with HIV, it will progress to AIDS-like symptoms. And then if you plot out in a phylogenetic tree the relationships between all the SIV lineages and then put in HIV and HIV1 and HIV2 in there, so there are two different forms of HIV found in humans, you get this relationship. And it turns out that HIV1 and HIV2 are not particularly closely related. HIV1 is closely related to SIV CPZ, or SIV found in chimpanzees. And HIV2 is found, or is closely related to SIV sooty mangabe, so SIV found in sooty mangabes. So these represent separate transmission events. And if you look closely at the relationships within even HIV1 and SIV CPZ, and then within this HIV2 SIV SM group, you can see there were multiple transmission events within these strains as well. And these transmission events have largely to do with the butchering and hunting of bushmeat. So we know exactly, because of those SIV strains, we know exactly where these transmission events occurred. So SIV CPZ jumped into humans in Cameroon, and SIV sooty mangabe jumped into humans in West Africa. So this is some work my colleagues and I did on these SIV strains, looking at SIV in West African red colobus monkeys and these red colobus monkeys in Kibale National Park, closer to East Africa or Central Africa. And it shows that they're closely related phylogenetically. So the simplest explanation of that is that they co-diverged with the host. Now, if you look at some of my earlier work on divergent states within the African colobines, these two populations, this West African red colobus monkey and these ones at Kibale, so some of these are the same individuals that were even sampled over here, diverged about 3 million years ago. So if you do have co-divergence between host and virus, which is the easiest way to explain this pattern that we see, it's likely that SIV has been around for a very long time. So what we know is many African primate species are natural reservoirs for SIV. SIV evolutionary history involved extensive host switching. So you have switching from not only non-human primates into humans, but also between different non-human primates. There's a lot of recombination in the SIV genome between these different SIV strains. And SIV likely has a long co-evolutionary history with African primates. So this is what we don't know. And this is what we're really kind of trying to figure out in this project. What factors affect SIV transmission and persistence within wild primate populations? So the kind of phylogenetic framework for SIV has been well worked out and been kind of done to death. But we really don't know anything about how SIV circulates within a population. So what are the internal population dynamics of SIV? How human changes to the environment affect these factors? And how human cultural attitudes towards non-human primates affect zoonotic transmission. So we know a little bit about what's going on in West Africa with bushmeat consumption, but we don't know much about what's going on in East Africa. So East Africa is a little bit of a different cultural situation. You don't have as much bushmeat hunting or bushmeat consumption because there's a larger Muslim population and there's a Muslim taboo against eating primate meat. But there are other instances where you could have transmission of these viruses. So for example, killing primates when they crop raid. There are examples of humans killing baboons or colobus monkeys and then chopping them up and feeding them to their dogs. So, so there are definite practices or behavioral practices that humans conduct that can expose them to these retroviruses. So here's the model system that we're looking at. You have Kibale National Park here in Uganda. And then here's the core forest over here. And what you have over here are different fragments in the unprotected area. And in between these fragments is agricultural land. So these fragments are created by human activity. And so what we want to try to figure out is, how is human alteration to this landscape affecting the demography of the animals? So how is it affecting migration throughout this metapopulation, throughout this matrix? And then how is that in turn affecting disease transmission? So we'll have, we're sequencing retroviral strains and looking at the relationships among these different forests, these different fragments. We're on the host side. I'll be looking at gene flow throughout here. So there will be kind of a network of disease transmission and network of migration throughout this matrix. And then in the local community also, we'll have some ethnographic and sociological work being done, some survey work being done to see what humans are most likely to engage in risky behaviors and what drives them to engage in risky behaviors in terms of being in contact with these animals, and then how retroviruses might spread into the local community from those individuals. So we'll have these various different networks of disease of the host animal and then of humans, and then ideally we'll have kind of contacts or these potential nodes where these networks kind of intersect to get an understanding of how exactly retroviruses are flowing throughout this community. And there's other aspects being done as well, looking at kinship within social groups. There's a behavioral ecology aspect looking at social behavior and how kinship and social behavior might also be affecting disease transmission. So that kind of outlines that project. Like I said, I have no results yet, but I'm in the process of hiring a postdoc now to work on this and really kind of taking a more genomic approach, moving away from microsatellites and mitochondrial DNA and looking, maybe using something like RAD-seq, and also developing methods where we can get genomic data SNPs out of fecal samples. So that's what we'll be doing over the next couple of years. So that kind of gives you an idea of what I have done and what I'm doing now, and what I plan to do in the future. But I'd like to kind of acknowledge these collaborators that have worked with me on these different projects, different funding sources that have funded my work. And I'll be glad to take any questions.
All right, we have plenty of time for questions for Nelson, but first I just want to mention that there's a reception in 327 Pacific, super done, just down the hall. Many of you have been at 327 Pacific. If you haven't been there before, just follow everybody else down there. And if there are any questions after you exhaust Nelson now, you can keep going at the reception.
Your last project, I assume that must have a large sociological component to it?
Yes. So there are multiple PIs on that project, and one of the major things that program, Ecology and Evolution Infectious Disease, likes to see is a kind of social science side of a project, and that integrates well with the biological side. So we did that by having, like I said, there'll be some ethnographic work and some survey work being done in the local communities that surround the forest to see, to interview people to see, okay, what instances of contact do you have with these primates? Why do you have those instances? What those instances were? What drives people into those situations? So, like I said, to try to figure out, what individuals are really at risk of contacting primates and contracting these retroviruses. So yeah, we have a bunch of collaborators in the UK who are epidemiologists, cultural anthropologists, and sociologists who will be doing that work. And then the idea is that they'll be integrated in using the kind of network analyses. Patrick?
So you showed that one paper with the improvements in the historical demography with 16 loci. What about 20,000 loci? Do you have any sense of how that will start to saturate, at what level, and how much power there can be in that kind of analysis?
Yeah, I don't have a great sense of that. I mean, I think as far as what point you get saturation. Yeah, that's a good question. I don't know if anyone has looked at that explicitly, using that methodology with the skyline plots. But I mean, it's very clear that a single marker is very limited in what you can infer. But what the kind of maximum is, it probably depends on. Clearly, if you use ones that are unlinked, that's going to be ideal. So you don't want to saturate to the point where you have markers that are in LD with one another or anything like that. But yeah, I'm not sure if people have tested that specific method, see what the upper limit is as far as multiple markers.
Is 20,000 as easy as 16?
Right. Yeah, I know. And especially, and that's why I'm really excited about using some of these new methods. And the type of work I've done has largely been constrained by these non-invasive sampling methods, and getting around that is something that I'm looking forward to doing, and something that that we're trying to do some elephant work with Susie Bassam in Bill's lab on that, and then hopefully that will be working in parallel with some of this colobus work as well. So I'm excited about that. Paul.
When you're dealing with the spread of disease between populations here, well, let me give you some examples. We have some evidence that baboons in East Africa the males can travel as far as 100 miles away or even more, which would carry, of course, disperse whatever they're dispersing.
Right.
And another case is the bonnet macaques that I studied, for example. I'm not sure how this holds up now, but the idea was that they were pretty inbred and did not disperse very far from their birthplace. Mm-hmm. And these are both animals that can move on the ground. Mm-hmm. I don't know that we have enough evidence for a lot of these forest-dwelling species about how far they actually can move and carry whatever they're carrying with them. Do you have any sense of that?
I mean, it's definitely, in terms of the kind of species that where males are primarily dispersing, it's definitely been shown that males disperse further than females. Even if females disperse, they'll probably just disperse to an adjacent social group where they might find kin. That's one of the hypotheses. As far as how far they'll disperse, I mean, I think if you look in the ethological record, I think for some species like capuchins, I think they've done some work on that where they kind of identify individuals moving a pretty far distance. Maybe some of these markedly captured genetic studies can do some of that, but I don't think, I mean, not like they've done with macaques and baboons, where the animals are really well habituated and can be followed long distances.
It's much easier to follow baboons than macaques.
Right. Yeah.
I'd like to follow up on that. I think this is actually really pretty fascinating to think about the network of connectedness of the host and the parasites in terms of the genetic relatedness. Because if you think about gene flow, from the host perspective, is not just how far you move, but then what's the fate of the individual once it gets there, whether it breeds or doesn't breed. But that's very different than, and that might actually, there might be some reasons why an individual might travel a long way and not be successful in mating, and thus gene flow might be less. That might be the opposite for irises, for example, where they encounter a naive host, for example, it might be increase the probability of transmission of the virus, even if there's an increase. So anyway, have you thought about that in terms of the sort of counteracting, counterprevailing processes that will affect the genetic structure of both the host and parasite?
So that's why we found this model so compelling, because basically you have these different fragments, by this core forest, and like I said, they're different sizes and different distances. So you have almost kind of an island biogeography thing going here, and like I said, we're not interested just in SIV, but other retroviruses as well. So we'll have a pretty good idea of we've already done some preliminary work on retrobiodiversity in these different fragments, and it's definitely not consistent. So why is that not consistent? Does it have to do something with a distance area type relationship? I mean, that's one of the things we'd like to test, to see, okay, is there some kind of balance between colonization and extinction? Does that have to do with the shape or distance of these fragments from the forest? And I mean, that's something that we'd like to test. I don't think we have any like I said, we don't have any results yet, but it's something that we're interested in looking at. Yeah.
All right, let's thank Nelson one more time. Don't forget